Okay, natively unstructured disordered proteins. Until now, in the lecture, I've always talked about how sequence determines structure and how structure determines function. So how this process of coding the information in the DNA leading to a particular structure leads to function, and that has to be that way. If anything goes wrong, function doesn't work. And the typical idea is sort of illustrated here by these two molecules, trypsin and its inhibitor. Trypsin in blue, the inhibitor in green. And you see this is really the idea of key and a keyhole. These two molecules fit together just like a key into a hole, and it can unlock, this key can unlock only one door. That's the same story here with trypsin and its inhibitor. Only those two molecules meet, only those two molecules fit. This fit, and you already see that in the keyhole, in this, and that's where the keyhole analogy breaks down. The keyhole analogy, you have two static objects that have absolutely no dynamics. So the locksmith has put all the intelligence in making these two things fit to such an extent that dynamics is not needed. It's a little bit different from modern locks, where you have to slide in the key, and then when you look at the detail of the mechanism, there's something about a sliding contributing to the mechanism. But in the world of proteins, this is really very, very, very different. It's motion that implies, and it should have started on its own. Let me see. No, it doesn't. I'm sorry. What did you say? It was moving. Okay. If it was moving, then we get it back. Oh, yeah. But this one doesn't. Oh, yes, it does. Okay, so you see here phosphorylation, you have a swing out of a longer part of the molecule on the left. While on the right, when you see the beginning of it again, there really are two different parts of the molecule that swing together and then bury the substrate. Triose in this particular case, you see the substrate comes and the whole protein wings over and buries it. So this entire thing is the binding process. Those two are on slightly different time scales, bless you. Those are two just examples for the binding of a molecule, and you clearly see this is dynamic. The process of binding is one that involves changes. And again, where the structure can change how, so this bending mechanism that you see on the left here, is one that is completely built into the structure. You can only do it in one way. The hinge works only in one way. Now, imagine you had a different story. And this is referred to in different words. I use mostly the word disordered, natively unstructured. So you have a stretch of a peptide, a protein. And this protein is natively unstructured or disordered. What it means is, if you could shine light on it and you could observe it through light, we cannot, they're too small and too mobile, right? But if you could, and you would shine light at two different time points, you would not see the same, because it wobbles around, okay? Upon binding to this molecule here, what you see is that regular structure is induced. So you see the formation, by now you recognize the helix here and another helix. So suddenly this previously wobbling, or on its own wobbling protein, has adopted two helices, and these helices are in place and stay in place. Now, in this conformation, if you now shine light at two different time points, you will always see the same. Now it's structured. And this is a mechanism that is on the scale of just, you need some motion. Here you need a little bit more motion. So there's a lot of motion. Much, much more than I showed you before. In these images, on these movies, what you saw is well-structured proteins that move around a little bit. Here you have much more motion. One mechanism in which we sort of imagine this is the analogy of fly casting. So you, at the end of a long line, you have a fly, you throw this with a rod, and with this mechanism you can reach out further. Then the analogy, the fly, the fish takes the bait. In the world of proteins, essentially a weak contact is formed. So the line sort of thrown around this one here, and there's a slight contact that is forming. And this slight contact then sort of pulls the rest in, and then you have an induction of, in this particular case, these two helices. That's the idea. So you do this in order, for instance, to increase the reach in which you can sort of attract. You do this also because, what else can you imagine? What could be another reason why this mechanism would have an advantage over the key lock mechanism? So one is reach, so you can reach further. What else could you imagine? Just look at it. So you throw this line at it, and it sort of sticks somewhere. And then something is induced. As opposed to the key lock. So you have a key that fits into a lock, and it opens one particular door. What can you imagine here?
Maybe it can take on different structures depending on the...
It can open different doors. And in the analogy of opening doors, right? So because maybe this glue, you can induce slightly different shapes into this glue. And maybe all you need is a glue that fits to the first contact, and then whatever you wheel in will find some place to sort of dock and bind closer. In fact, that is one of the main reasons we see for the existence of this natively unstructured regions or disorder. There are different types, and so this slide here shows is from a review that is now 13 years old, but it shows different types of disorder. So what I just said is one type. So you can bind to different proteins with the same protein. So the same protein is now you have a key that works in many doors. So that's one idea. The other idea is, again, this reaching further out. So opening many doors and reaching further. Now this reaching further has another aspect. So you could imagine that when you reach out, you can actually also sense further. And that again is what exactly disorder proteins do. So you increase the width, the reach that you have by just stretching out instead of having a well-structured protein, and you sense your environment. And then there is some reaction if you sense that something is coming into an environment. There is another mechanism in which you actually prevent things from coming. So it's like a distance holder or whatever you call that. There's a third mechanism, or fourth or fifth or whatever you count. Another mechanism that has been observed, which is what many of you, when you order your packages from Amazon or something like that, you get filling material. And this filling material buffers. And this again, so proteins that remain disordered, they never adopt one of these structures. You said they could adopt two different structures to bind to two different molecules. Yes, but there are some of those who we have never observed adopting any regular structure. And we assume, or in some of these cases at least, the assumption is that they function like filling material. By never really being any real structure, they never have a binding. They never bind to anything. So it's just the opposite of binding to many things. It's also disordered, but it's preventing from binding. And it is put around proteins to sort of avoid that others can intrude and bind. So blocking of wrong connections. Now there's different types of disorder, I said, in terms of what it does. There's also different types in the way we actually predict it. The simplest way in which you can sort of approach this problem is what I call here loopy disorder. So what we observed when we analyzed, when we used our secondary structure prediction method, and we analyzed entirely sequenced organisms, we saw that many proteins have long regions in which we do not see any regular structure. So regular secondary structure is helices and strands. If over 70 residues, as this formula on the whiteboard gives you, over 70 consecutive residues, you have less than 5% helix. This is something that is very unusual. So I've analyzed proteins for many years, and I'd never seen anything like that. So my immediate reaction was, this is something strange. And in fact, my immediate reaction was, this is something so strange, this cannot be, must be a mistake. So I went back to look for the mistake, and then we sort of more systematically found some of these cases. Then we wanted to find out what could it be. So then you try to Google around, what are these cases, and you find nothing. So the next best thing you can do is go for proteins for which you know the structure and look for something like this. Doesn't exist in the protein structure database, or didn't exist when we looked at it, so we had to decrease the number. So we did the same thing, sort of 5%, but 50 residues. That we found in the structure. And we found sort of 4 different ways of doing that in the PDB. So proteins of known structure, again now it's less than 50, but still over 50 residues, no regular secondary structure. The 4 types of cases we found. One is connecting loop. So you have this essentially 3 domains, 1 domain, 2 domain, 3 domains here, or you can count it differently. And then there is something here that is connecting these 2 domains, a long loop. And this long loop happens to be just 50 residues long. That's one example. It's not quite disordered, it's just one floppy region. And there was another example here where you really had at the end of protein, and we found many of those cases, where you had floppy ends, and they could be up to 50 residues long. Floppy ends again means there's no regular secondary structure, or less than 5% regular structure over 50 residues. The third one we found, the third type of case, and overall we found, I don't know, tens of cases. The third one is where the loop, similar to this one, but now the loop is actually not connecting to domains. Now the loop is sort of wrapping around the molecule. I don't know, like a wrapping paper or something like that. If you look at the spatial film model of this, you see it nicely fits in there. And the fourth kind of situation we found was like this one here, where the way we thought about it is if you took it and you compressed it in all directions, like a snowball that you press, then it would look like a regular structure. But the way we saw it, there were no bonds formed. So there were very few beta strands, there was very little secondary structure because everything was too far away. It was as if you had a regular protein, you sort of blew it up. And then it cannot exist because it doesn't connect, right? And some of these cases look like this. This particular case here, we published that, and this particular case turned out later to be an experimental mistake. But there are cases that are a little bit like that. So this is not the only one, we just chose this one. And then we looked at how often do we find this sort of thing. Now we get back to the 70 residues. How often do we, since we can predict secondary structure for every single protein entirely sequence organism, at the time we ran all the known organisms. At the time that was a list of about 30. So for 30 different organisms we asked what fraction has at least one region of 70 and more residues that have no regular secondary structure. And what fraction of residues are presented. So on average, the average is not here, because average doesn't make sense, but let's just look at let's look at this one because it's easy to see the number. You see that about 13% of all the proteins have one long region, and these long regions amount to 7% of all the residues in this entire organism. Now, 7% of all the residues, 20% of all the proteins, this is not garbage. So what we're looking at here is clearly not something that is just a coincidence. There is some functional reason. It's a lot of residues, 5%. And you see another thing here. Up where the bars are much higher is where the eukaryotes are. Down here is prokaryotes, so that's bacteria and archaea. And you see that eukaryotes are way higher. On Tuesday, I briefly showed you that the number of membrane proteins are similar between eukaryotes and prokaryotes. That the number of coil long helices are similar. And there are many, many, many things that are similar. Here we have something in which they behave very, very differently. In fact, when we looked further by length, a cumulative plot by length, we saw that this is overall we found 50%. And that's longer than 70, when we asked how many are longer than 110 or something like that, we still found 5%. In the cumulative percentage, 20% of all those that we found, in fact, were longer than 170. So many of these regions are extremely unusual. Why is that? And how else could we? Well, what we saw is that they correlated with what other people had developed as prediction methods. They looked like they really captured disorder. There's another way. If this order has to do something with movement, with motion, then you assume, if you predicted motion from sequence, such a thing as the b-values, some of you may remember in the exercise in the beginning you sort of looked at b-values. Again, b-values give you an idea about how much motion every residue is under, or how much it can move. And I also showed you examples of NMR structures, such as this one here, where you see many different structures for the same protein here, because you have many different lines. And then you have points where all these different lines come together, or regions of points. So these helices here in the middle, they are so black because they're very well defined, well defined in the sense that all models fit at that point. And that is interpreted to mean that way the blacker it is here, the less motion. The more they differ, these different models, or different structures, the more you assume that at that particular position you have motion. Overall, over some data set, you can sort of normalize the b-values, get a distribution like this. In this direction you have more flexibility, in this direction you have more rigidity, and then you somehow can introduce a threshold to distinguish in the binary state flexible from not flexible. Sometimes in exams we ask the question, where would you put the threshold? So if you want to carve this is the raw value, the raw distribution. If you do machine learning, you may try to in fact learn this raw value. And it turns out this is not that easy. So it turns out that there are good reasons why you want to sort of try to predict a binary value. Again, the binary value is flexible or rigid. Now the question is, how do you translate this distribution into binary values? Where is the threshold? And many people would put the threshold into the peak. Why is that a bad idea?
Because that doesn't seem like a Gaussian distribution, it's a little skewed.
So the distribution is skewed, it's not a Gaussian distribution, but still this is the peak. So this is not an argument for not using the peak. So would you do it if it were Gaussian?
Yes.
Okay, so why is it a bad idea to put the threshold into a peak of a distribution if it were a Gaussian? Just imagine you see a Gaussian. You don't see a Gaussian, completely correct. But assume this would be a Gaussian. Why would it still be a bad idea to put the peak for distinguishing what is binary, so on one side of the Gaussian and the other side of the Gaussian, right into the average, or into the peak? Yeah?
We know that most of the data is lies between 1 sigma, 2 sigma, and 3 sigma.
Yeah.
Peak. So putting it right in the middle would kind of, I don't know.
Yeah, that's the problem. So far you get a point. This question gets 3 points. The 2 points are missing are the part where you say, I don't know. Nail it. So in the examinations, I believe you're onto something. Just think it through. Just spend a moment on thinking it. Or there was somebody else? There was somebody here before. Was it you or no?
I don't know if you put in the peak, in Gaussian, you're just taking 50-50.
Yes, that's correct. And?
Yeah, but it's like informative.
Well, yes, it is informative. In fact, for machine learning, you could even say this is the ideal distribution. If you have 50-50, you can learn both classes equally well somehow. So this is the counter argument. This is, in fact, most likely would be the argument why you would want to put it into. It could be one. This could be used as an argument to put it into the center. Your argument is right. You just have to turn it upside down.
But if there isn't a, sounds like a really scale. Like you would have 50-50.
It sounds, because I say it isn't, but my question is why? Yeah? No. Think about the epsilon. You all have done enough math to know how people typically use the epsilon. Go sit yourself on a peak and think about an epsilon. And think about error. Somebody?
What about an upper and the lower bound?
Yes, so this is somehow what we, in fact, we have two thresholds, and it's a little bit like that. Because we could not, so we have a solution, we're not quite happy with the solution, we do something like upper and lower bound. But the issue with the peak is the one that I still like to address. So what you have begun to say, think if the peak would be the solution, ultimately this is not a difference between this being a Gaussian or this distribution that I have here. The problem with the peak is every experiment makes some error. Okay, this measurement is never 100% right. There's a tiny epsilon of a mistake. If you put your threshold into the peak, the tiniest mistake has the strongest possible effect. Because in epsilon plus minus, by definition, if you're on the peak, it's the strongest effect. It's the largest part of the distribution that sits around this peak. So any deviation, any error has the strongest effect if you choose the peak. That's the problem with the peak. If you had values, you never had, but if you had values are completely error-free, we could rediscuss the story. But given this problem with the error, you have the maximal number of samples that you classify as one side of the peak, although it should have been on the other. So any epsilon, any error margin that you have, by choosing the peak, you will always maximize the problem that you will create for the machine learning device. That is the problem with the peak. So in this context, what you would like to do is some sort of extreme values. So something where epsilon differences do not hit many points. So ideally something like this or that. That doesn't work, because then if you put the threshold somewhere here, you just don't have enough points left. The entire distribution sits on this side, or you put it here, the entire distribution sits on that side. Doesn't work either. In that sense, the idea of having an upper bound and a lower bound, or having different thresholds, begins to pop up. But one thing, there is no right solution here. What you always should try is to put thresholds, if you need a threshold, and again, there are reasons here why we absolutely need a threshold, put the threshold somewhere where the threshold has less effect. You see that the two thresholds we put here are both still in the relatively extreme part of the distribution. This in particular here is a little bit less so. So this was exactly chosen because there's a little bump in the peak. But you know, you cannot solve this problem optimally, but just choosing the peak is the optimal wrong solution. Everything else may not be good, but it is already better than the peak. Okay, now then you can develop whatever the peak is, or whatever the threshold for the distinction is. You can develop a method that predicts just the same way we did that before. You use profiles input. Everything as I described it for second-order structure prediction, except for this particular case, you don't have a second level. You only have the level that takes the sequence of input, that's all. And you can predict B values here running along the protein. High B values in this particular case relates to high variation, so these are very rigid molecules. In fact, these three that are marked, shown in the structure here, are the ones that are on the binding side. Here we looked at two examples, and in these two examples we, in fact, in one case for Rost, we could show the conserved part that had to be flexible for function, and it was conserved, and the other one we could show the conserved part that had to be rigid. So the method could distinguish between important sites that are both, that have to be really flexible and that have to be really rigid, and that's why it also made it through, this picture made it to a cover of a journal. By the way, this is something else as an advice to you. When you do your thesis, when you do a paper or begin to write a manuscript, when you sell any story that you want to sell, try to sell it with an image. Try to think about an image that is easy and that really is something where you feel this should actually be somewhere on the cover. This should be somewhere when I enter my building. That's where that image should be. Think about these types of images. Typically, when you write your thesis, when you write your manuscript, the figures you do are not of that type. They typically have a level of complexity that evolves this way. You dig deep into understanding something, and when you make the figure, then typically it's rather complex. And then somebody criticizes something, and that makes the figure more complex. Somebody else criticizes, the figure gets increasingly complex, and it has to be that way. Just find the moment to step away and think big, and think about how can you simplify your finding in a simple figure. And if you have such a figure, spend time on it. It's really worth it. Whatever you are after, it will give you more money, it will give you more fame, whatever, it will give you more happiness, whatever it is you seek in life. Try to think about this. It's really important. Okay. Yeah?
Can we go like to the spin of the, like this one? Is it, like, is there a reason why the increases and decreases like this motion with that?
So is there a reason he asks why we have a particular shape here? We have a particular shape that you can describe as deep valleys, 1, 2, 3, 4, 5, 6, 7 deep valleys, and 3 at least of these 7 I said are important binding sites. So the question is, could I have predicted this shape, and will all proteins look like that? No, they will not. They look very different, but you also ask, is there a reason? Yes, there is a reason, because ultimately there is some repetition of flexibility. Some residues are more flexible than others. You will have something that will have ups and downs, and you will have some sort of theme that you can recognize as something, just because there's regularity built in. But the way it looks, the depth of the valley, the number of valleys, how far a valley is from valley, or whatever you think about what you recognize in this image, it's not predictable. It is specific to one particular protein, and I assume that if you look at these and you could compare 2 of these, you would recognize similarities. But that's where it ends.
So could I say that every flexible area or region needs a rigid area around, like otherwise, like you showed?
Well, so the question is, does every rigid area need a flexible region around it? You have to be careful. What I'm showing are differences. So the value you get, even if there's a tiny difference between very rigid and still rigid, right? You still get a value. It may not. So it depends on what part of it you mean. If you meant the shape, then you will get that even if almost everything is rigid, because some will be less rigid. If you mean with what you see here with the value, so are always all going that deep and some that high? No, that not. But there is sort of a but in here. We had to, the experimental B values that we get are specific for one particular protein. In order to have machine learning, we have to find something that is comparable between all proteins. And that means we have to introduce some weighing or some normalization. And that some normalization sort of brings a little bit the idea that you will somehow see it because it's enforced. But it is true, and the image here shows it. It is true that we can show that one protein is more rigid than another. So our prediction method is not always creating the same average. It's not always creating ups and downs to the full extreme. It can show cases where, in fact, almost everything is rigid. Okay? Now, the important point is, then we learned that this disorder helps us maybe, but it is not good enough to pinpoint disorder. I cannot say I find a region that is 70 residues long, that is flexible, and then I have disorder. I could say that, but we didn't find them. So that turned out to not really be, possibly because the prediction method is not good enough. Possibly because the prediction method exactly is normalized, because we enforce that this in fact sort of unlikely to happen. But anyway, so it did not quite work for that. So we turned back to the other method we had, the method that in fact predicts non-regular secondary structure over 70 residues. The problem with that method is it works, but we want to find disorder that is only 30 residues long. And by definition here, with 70, I cannot find, with that method, I can never find anything that is 30 residues long. At least not anything that is disordered, because it has to be more than 70. Now the simplest solution would be to just swap the 70 and make it 30. The problem is, then this method finds all kinds of things that are just the regular order. That's absolutely wrong. So it finds too many wrong ones. Okay, we cannot. So the 70 has been chosen such that we did not find any example looking at it where we immediately said this cannot be. In fact, we didn't find anything in 70 in the database of known structures. There was another encouragement for us. So for anything that we know the structure of, we could not find anything with that method. And that sort of helped us to feel confident that it produces something right. But again, I want to now apply this method to finding things that are much shorter. I want to somehow, this is what I have. Do you have any idea how you could sort of use this to create a machine learning data set to now train that you find something like that for short regions? But you had a question before.
So you're using the absolute value to decide, right?
Yes.
Why don't you use like a relative to the length of the protein?
A relative to the length of the protein?
Yes.
Oh, so the feeling is, let's think this through. So relative to the length of the protein, then the logic is that a protein of 100 residues would have a different region that is disordered than a protein of 1,000 residues. But that is not our intuition. Because our intuition, a protein of 1,000 residues is just built up by 10 proteins of 100 residues. I mean, it's an overstatement of the case, but you stack up these very long proteins by combining different domains. Okay? And each domain then looks like a little protein. That means within each protein, essentially, that length should count. Now you could still apply your argument. You could still say, okay, maybe not the length of the whole protein, but the length of the domain at least. Why don't I scale that with the length of the domain? And again, so the way you think about this fly casting idea. The fly casting idea works only if you have a certain length. 70 certainly is enough for fly casting. And if you have a shorter domain, you still need the line being 50 to 70 residues long, right? If you want to do fly casting, you need a certain length. And that is independent, this idea of fly casting, or the idea of sensing in the environment, or the idea even of filling material. All of these ideas are somehow independent of what the entire protein around you is doing and how long it is. It's more relevant that there's one region that can do that, that is sort of has the capability of being the line for fly casting, right? So this is why we really look at an absolute value. Again, the question is, this produces something that I know is disordered. I, on the other hand, have proteins that I know are well ordered. And now I want to have a machine learning device that finds things like that, but much shorter. How could you do it? Any ideas? How to create your data sets? Yeah?
Can you predict the structure regions of the protein?
Ah, so you can, again, you mean the secondary structure? Yeah, we can. Then, but you cannot make the number smaller. So you want something that in fact does not have regular secondary structure for 30 residues, but just looking for everything that doesn't have regular secondary structure for 30 residues and not more than 30 residues, you find many things where just it actually has secondary structure and you just made a tiny prediction mistake and that breaks this number down. And you make too many, if you have too many false positives. You cannot move this threshold to 3. But you still want to sort of find things that are only 30 residues long. So you hope that there is some common feature in these here, and that this common feature is what you can learn, and then it could help you in the machine learning device to find things that are 30 residues long. How could you build that machine learning device? So you need 2 data sets that you want to take apart. What are these? How can you produce them? Okay. So the true cases are the ones that you predict. So everything, you run this everything that is longer than 70 has no regular secondary structure for a large set of proteins. We ran it on 30 entirely sequenced organisms. And with that, we predicted everything that fits to that number, larger than 70. And it's the false ones, the ones that are not disordered, that are well ordered. We took the entire PDB, the proteins of known structure, because every one of them is well structured in our eyes, right? And we simply try to separate these two data sets. We try to see what is in common in these and what is in common in these, the well-structured ones. Mind you, there are,
Do we look at entire proteins in this case? So we say this protein is true, this one is false, or do we look at...
No, in this particular case, so the true part here is only the region. We take the region that is predicted, no matter what it is. I showed you a brief length distribution, a cumulative length distribution. So some of those are over 170 residues long, and then whatever is predicted as disorder is taken. That's the positive. And the negatives are not sampled from the rest of the protein, but the negatives are only sampled from different proteins here. Now, the sort of strange thing about it is an unusual way of doing machine learning, because the set of true contains not a single example of what we actually want to learn. What we want to learn are things that are shorter than 30. Everything in my data set is longer than 70. Okay? So what I'm actually hoping to get out is not in my true set. The other extreme end here, the false, the negatives, are the proteins that are well structured, the PDB. And I will briefly mention in a moment, there is a lot in there that is actually disordered. So in training this device, make many mistakes. My data set, my training set already contains mistakes, and nevertheless, it works like a charm. So this device can learn to predict Norse-like loopy things of 30 residues, and can distinguish between 30 residue loops that are disordered and 30 residue loops that are regular structure. How can it do that? And the trick ultimately here is that the only thing that is consistent is the signal. Yes, I put a lot of mistakes into my truths and into my false samples. But where the mistakes are, there is no consistency. The only consistency is the signal that I want to capture, and I capture that signal, and through that, in fact, I can distinguish between 30 residues loop disordered and not. It's a beautiful example of a method where you use a sort of simulated, and somebody asked that at some point, can we use simulated data set? This is one of the only examples that I'm aware of where we somehow use something that you may call simulated data. It's not really simulated, it's predicted, and it's lucky that, in fact, the real thing that we were looking for was in there. We were not sure about it. But this is one example in which it works. Here's another way in which you can do it. And the other way, sorry, I changed that. No, I don't. So the other way, yeah?
So the only thing consistent is the signal. What signal do you need?
So the signal ultimately here is that in the data set of the things that are 70 and longer, there is something that has to do with loopy disorder. There's some signal for how loopy disorder looks on the level of the sequence in terms of its conservation, in terms of its solvent accessibility, and all of that. That is the consistent signal. They have something in common. And this something in common is in fact exactly what determines even loops that are shorter than 30, whether they are disordered, or whether, again, we have two loops, we have two regions here. One is predicted to be 30 residues and essentially have no regular secondary structure. The other one is predicted to be 30 residues and has no regular secondary structure. This is predicted to look just like a normal loop in a normally regulated structure, and this is predicted to be like a disordered region. And it can take these apart because there are different features. And that is the signal that is consistent. And I'm not able to actually go further. So I'm not able to say this is because the lysine is conserved to 55% and has a little bit of an exposure prediction. I haven't looked at the signal on that level, so I cannot sort of tell you the rules. We have never tried it, and I'm not sure. If we tried it, we found rules that are easily understandable. Or helpful, let's put it like that. Okay, so the next way of approaching to predict disorder comes from Shoshana Dostiani and Isvan Shimon, and their idea was that disorder clearly means something you flop around. If you flop around, well, let's turn it around. If you are well connected, you cannot flop. So floppy means that you don't have a lot of constraints. Floppy means that this part of the protein that flips around, the moment you're bound here, this can no longer move, right? It has to be contact-free. So let's just see what are propensities of residues to form contact. So let's define the propensity for every single amino acid. Then let's look at a running window of, say, 30 consecutive residues and sum what's the contact propensity for these 30 adjacent residues. And if they together have a very low contact propensity, so they are very unlikely to form a lot of contacts, then maybe those will be disordered regions. They're not very constrained. They will not form a lot of contacts, as opposed to 30 consecutive residues that have a high contact propensity, right? And that is their method. It's called IDP. And I'm not, my slides are not quite good. For that, IDP comes later and only in passing. Now, when we approach this is one of the best methods to predict disorder that exists, if not the best. When we approached this, we felt that what is wrong with IDP is it measures a generic propensity. So the generic propensity, whether this region wants to bind another region, also has to do with whether the other region is there. So I told you that beta strands pair. Well, if the pairing partner is not in that protein, this thing here will not have a contact propensity, even if it has a propensity to form a beta strand, right? So we tried to see how much better could we do if we had a contact map predicted. And this is something that in this lecture I will not get to. So we developed a method, machine learning based method, that predicts 2D structure. Shown here is the residue number, one protein against itself. Dark entries here mean that these two residues are predicted to be close to each other, in contact. There's a lot of dark here, and a lot of dark. In fact, this is not looking like a typical protein. A typical protein, you would have something that has a ballpark of 10% black here. This looks inverted. So this is 10% white. Meaning we predict that 10% of the residues don't have contacts. Well, we should have predicted that 10% of the residues do have contact. So the method is not very good. At the time at which we developed it, it was the best available method. Today, there are some better methods available in some situations. So there is still no generic way of doing the contact prediction. So today, this is still not much better than that, which is why we cannot predict 3D structures. And for some proteins, we can predict contact maps. People can predict contact maps much better than this. Some proteins is about, I'm not quite clear about the number in human, for a few hundred to maybe 1,000 proteins in human. Contact map can be predicted so well that people can actually predict 3D structures for those. At this point, we have 20,000 proteins. Those are still the exceptions, but for some we can do it. For all the others, we still have contact maps that are not much better than this. But we felt that nevertheless, the contact map may be good enough to distinguish between disorder and not. And we had two proteins here. This is well ordered, and this is the predicted contact propensity, and this is disordered. In this particular case here, more contact, fewer contacts. In this protein, you see the red one here, most of the protein sits on the side of having very few contacts. So we simply developed a method that used these predictions of contacts by introducing some threshold, and if the threshold applied, we said and this is the threshold shown here, running the sequence. If it is below some threshold, we call it disorder. And here's an example where you somehow see that disorder is correctly predicted. Then when we analyze what type of disorder, we saw that the loopy we talked about and some other disorder differs from this contact disorder. And I'm not going into details. But why is this disorder? You see two nice helices. Why are they called disorder? Well, the point is actually these two nice helices, they are wrapping around each other and they bind each other, in this particular case, only 3 different positions through very weak points. Any movement of the environment, any change in the environment can disrupt that binding and essentially take these two helices away from each other. In that sense, it behaves a little disorder-like because there is a difference at 2 different time points at which you shine light, and in fact these helices slightly unwind. In this particular case for the Max transcription factor, this slight unwinding is in fact part of its action of binding DNA. Now, what I want to point out at this point, I've talked about a few machine learning devices targeting to predict disorder. Not a single one of those used data about what disorder actually is. And the reason for that is we don't really have a clear experiment that shows disorder. There's nothing that really says measure disorder. What we can sort of measure, and that goes back to something Keith Dunker introduced, what we measure is, let's get back to this slide, when we look at 3-dimensional structures, there are parts in these 3-dimensional structures that we cannot see, regions that are not visible. And the argument from Keith was, maybe they're not visible because they're disordered. And that's in fact how Keith Dunker from Indiana University completely introduced this concept of disorder. And he ran around for many years trying to convince people that this is an important part, and finally has succeeded. And he was always right. This is a database that exists. With this database you can do a meta-disorder predictor, and I'm going to flip over that. It's not really relevant. And I'm going to flip over those two. Ultimately what they say is that disorder is in fact more difficult to conserve than regular secondary structure. So regular secondary structure is less sensitive to random changes than disorder. So disorder, although it sounds disordered and not something that is special, is extremely difficult to maintain. An evolutionary set, bless you. So random hits against it, that's one thing. Is disorder now a stepping stone in complexity? And this argument comes, so here are 2 different prediction methods, IUPRET and another one I didn't talk about. And what you see, eukaryotes have just much more disorder, factor of 3 here for both methods, than prokaryotes. So what we know is that eukaryotes have more disorder than prokaryotes. Eukaryotes are more complex than prokaryotes. Maybe then disorder has to do with complexity. In fact, if we go further, multicellular, so eukaryotic organisms with more than one cell, eukaryotes have more disorder than unicellular, so ones with only one cell. We know that mammals have more than non-mammals. And we know that humans have more. So somehow this fits our feeling of we are on the top of some pyramid, and disorder relates to complexity. One mechanism by which this could happen is the so-called molecular recognition element, MORS. This slide comes from Keith again, where you again, like you have 2 proteins that you can bind, you have the possibility to sort of connect things in very different ways with these disordered regions. The next slide. Don't try to understand anything on this slide. This is one of those counter-examples of what I recommended to you. Find a simple slide that everybody understands. We found a slide that nobody understands. Let me just tell you what we did. And it's unfortunately really complicated to find a better slide. Or put it differently, I have so far failed. What we looked at is, when we look at organisms that live under extreme environments, so there are organisms that live in thermal vents at very high temperatures. There are organisms that live in the extreme cold. There are organisms that live in very salty environments. There are organisms that are exposed to a lot of radiation. Those would be extremely, and these kind of conditions kill off almost anything that meets them, and then there are some specialists that survive in these conditions. And the question was, if I look at this organism and I simply ask, what fraction of the proteins has disorder? Could I find that organisms that live in the same environment, in the same habitat, are similar in terms of their disorder and dissimilar from their closest evolutionary relative? So I could compare a particular pair of organisms that are in the same habitat, bless you, and for one of these two here, I could just find its closest neighbor or its closest relative and ask what is the disorder of the closest relative. And what the figure says is the closest relative are less related in terms of disorder than the ones related in the habitat. Meaning that if you just add or subtract disorder from your proteins, you can sort of survive as an organism in a different environment. And that's incredible. At least this is the way we see a correlation. We see the correlation in terms of numbers. We don't have the causative. There are some experiments that have suggested that in fact you can really do that, but so far nobody has really engineered an organism that can live in the cold by just adding disorder. So that level we haven't reached. Here's another story that is outstanding biology, I felt, from Ona Dahan and Yitzhak Pilpel. So they look at many generations of yeast. So many for them is thousands, many thousand generations. And then, like every good molecular biologist, they mistreat their cells. So in this particular case, mistreatment is you expose them to heat. And the heat is so high that essentially most of yeast dies. Some survive. And when you see, then you ask yourself, the ones that survive, why do they survive? What is different between them and the ones that don't survive? And it turns out that the ones that survive, in fact, all duplicate an entire chromosome. Yeast has 16 chromosomes, and one of those is always duplicated. So chromosomes is just a way to organize your proteins. Yeast has 6,000 proteins, and one chromosome, 6,000 by 16, in this particular case, has about 150 proteins, the chromosome that is duplicated. So it's the 150 proteins that are duplicated. Now, why would you duplicate these 150 proteins? You may argue, well, on these 150 proteins, you have something that helps you to survive heat. If that is the case, nobody has found that yet. The only thing we see here is that there's a correlation. So this chromosome that is duplicated, if I simply chose the chromosome with the least disorder, then that's exactly the one that is duplicated. I'm not explaining anything. There's no causative statement. There's a very simple organism that explains the reality. That's the only thing out there that explains the reality. Why that is the case? Well, again, from the analysis of the heat shock surviving or proteins, organisms that survive the heat, we know that if you want to survive the heat, you sort of reduce disorder. So in that sense, it makes sense. But duplicating a chromosome, I mean, you have the ones with a long disorder already there, so duplicating is unclear. Anyway, so you increase the content of proteins with less disorder. That's what they do, and that's what is successful. And only after about 1,000 to 1,500 generations do they really change the molecular biology. And really, that's the point when proteins that help you against heat take over. That's the moment after 1,000 to 1,100 generations, 500 generations. So back in terms of people, as Homo sapiens, we have not reached that number. So that's when if at the beginning of Homo sapiens somebody had put a hammer with a heat, like these experimentalists have done here, then we still would be in the emergency of gene replication part of this experiment, right? And then again, after 1,500 generation, there's sort of a change of the organism, of the surviving organisms. Now, a final word with a term coined by Sean and Lanague in Sydney, the dark proteome. And I'm going to skip over the part of the light in the proteome. The dark proteome here are the people. Dark, the idea is, assume you have a structure, this is a particular protein, and assume that the part that is dark here, you know nothing about its structure. And that is what is called the dark proteome. what fraction of our proteins are such that I can neither do comparative modeling nor have an experimental structure. So I have no idea how to look at 3D structure. And the answer for eukaryotes, bacteria, archaea, viruses is that there are sort of 15% dark proteins, 20 additional roughly 30% dark regions, of proteins with dark regions, and then there's sort of gray, and then there gray means I can build models, and then there is the one where I really have structures. So the next question then would be, why? Why is there such a lot that is dark? Is that because we haven't done enough experiments? So I said we have 120,000 protein structures, 120 million protein sequences. Maybe that's the simple explanation. Maybe in 5 years this number will go down. Or is there something out there, maybe they are different. Maybe they're new kind of proteins, and they're the kind of proteins that we cannot do structures for. Well, maybe, for instance, they are disordered. I talked about briefly mentioned that membrane proteins are very difficult to determine structures for. So let's just look at these sort of common suspects for why we may not have any light or any structure. The data set now 100% is dark, all the dark proteins. Okay? And you see some fraction of it is disordered. Some fraction of it is transmembrane, and compositional bias is also something that's difficult to do structures for. And in that sense here, you would say roughly 50% of the eukaryotes up here are such that I cannot really explain why they are dark. For half of it, I can. My question to you, why is that? This is published. And I can attack this publication because I'm even a co-author on it. This is wrong. What's wrong in here? So the statement is that 50% is unexplained, but sort of 50% I can explain. Is either disordered, we know we cannot do structure for it, membrane, difficult to get structure for it. We learned that already in this course. What's wrong in this explanation?
So what's meant by that, like this is dark protein? Like, does that mean that I don't know all of the...
yes, so what means dark protein means that there's no part of this protein for which I have any idea about its structure. So this is done here for a subset of the proteins, just to make sure that I get a clean data set. I look at the subset of proteins for which nothing in terms of structure is known. Okay? So what's wrong in what I showed you? My argument is 50% or more than 50%, something like that, plus minus. It's not about error bars here. It's not about plus minus something. There's something much more fundamentally wrong with this statement here. The argument is because 50% I sort of can explain disordered composition bias and transmembrane. 50% is remaining, and those 50% is sort of unknown. Maybe they will be done, maybe that is new proteins. What's wrong with this argument?
Well, you just assume they are ordered globular or low-count bias.
Yeah.
You cannot say anything about it. You don't know anything about the structure.
That's true.
It could be something very different.
Yes. No, so he says, I should not write these things in here. Yeah, that's absolutely right. So what it means is that the gray part is not explained with the right. That's all. And I do not know what it means. In fact, the question remains what it means. So is it something that I have never seen before, or is it something that in 5 years from now will be done? I do not know that, and I do not want to claim that. But the analysis is wrong. I say 50% I cannot make a statement about, and I look at the dark proteins alone. That is the mistake. What I should look at is the same prediction method for another data set, which is known. So I should look at a subset of proteins that are non-dark, for which I know the three-dimensional structure, and apply the same sort of prediction methods. And when I apply the same prediction method, you see that the ones that are non-dark, they also have disordered regions. They also have transmembrane regions. And in fact, I should at least subtract whatever the number is here maybe that is 25%, no, it's more than that, it's 30%. And then I have to recompute. So I have to turn this remaining 70% into 100. So I have to multiply with 1 divided by 0.7. And then compute what is that fraction. And that is a very different number than 50%. Now the question remains, what is it in this 50%? Is it anything that is biologically reasonable, important? So we have just submitted yesterday, I hope, a paper in which we, Andrea Schaffhans and John Ranagio, in which we show that when we look at the expression means how many of these proteins do we have in an actual living cell. The expression increases in this direction, and that's just the density. The dark proteins are a little bit here in low expression, a little bit here in high expression. The white protein parts are here. The gray is where white and black are overlapping. Most of it is gray. Most of it is such that, in fact, what you see here is dark proteins behave like non-dark proteins. There's no difference. So they're tiny sort of bumps here. Most likely they're statistically not meaningful. Now that means that at least what we can say about the dark proteins is they are relevant for function, which doesn't tell us whether they have any new principle. How would you sort of attack the question of whether they have new principles? What would you try to do? So, in one way is you wait 50 years, 100 years, and see whether any of those is going to be done. If they all will be done with today's techniques, they did not have anything new. Downside to that method, it will take a while. What could you do today? Not much. Yeah?
Maybe try to do like some clustering on the...
So you could try to do some clustering. So somehow see, is there something in common for dark proteins? The most extreme, can you machine learn to predict what a dark protein will be? So can you predict dark proteins? So this is a way of analyzing dark proteins, but could you actually predict them from sequence? Directly. Is there something in the sequence that in fact tells you that whether a protein is dark or not? If you can, and what this one shows, the difference between the green and the red means that somehow we can. If you can, then there at least is some signal, and we are currently trying to see how much of a signal there is. And yes?
Why would you want to predict like dark proteins?
Because my hope is, so why would you want to predict a dark protein? So my hope is, say half of the proteins today in my classification are dark. So what I hope is that there are some in there that I can find an experimental colleague and tell them, listen, those, they are dark because nobody has really worked on them. Do them. You can get a structure. And then there are some, they are really new. And I give them to different colleagues and say, well, these are going to be tough for you. But if you can do them, you will find new principles. That's why.
In the future, that means also like with more knowledge, like some of those like dark proteins will become white.
So we hope that some of these dark proteins will become white. So again, the prediction method could help you to distinguish the ones that become white and that will not become white. And it could possibly also distinguish or help you in the way you design the next experiment. The ones that we predict are really the bad dark type. Do different experiments. So first figure out what they do in function, because getting the structure for those may be impossible. And maybe do a little bit more, yes?
This might be a question about the classification of dark proteins. These are proteins that don't have comparative modeling doesn't work for.
Yes.
And things that people try really hard to do, but can't.
Yes. So what are dark proteins? Again, those are proteins for which we don't have an experimental structure despite, well, actually not despite trying. We don't know why. Maybe because people tried, so we cannot really see the difference whether people tried or not and didn't get them, or whether nobody tried. And we cannot build a comparative model. Yes. That's right. Yeah? So what we have as information is we have the proteins that are currently labeled. It's a little bit like the story that I showed you before. We have the information about the proteins that are labeled to be dark today, so they are known. Okay? And then we have the set of proteins that we know are not dark. And we try to put these two data sets apart. Just like I showed you with this Norse net kind of method. I hope this, so it seems that it sort of works. Any other question?